Welcome everybody to the 25th webinar of Instruct-ERIC Structure Meets Function. This time it's the opportunity to have this webinar presented by the Czech Center of Instruct-ERIC. The Czech Center, you might know, is actually two-headed. There's one center in Brno at CETEC Masaryk University and another part of the center providing services in Bioserv Investors near Prague. And today we have two speakers, one from Brno and one from Prague, actually. We'll start with Tibor Fuzik. Tibor, who is currently at Pavel Plevka's group, the Laboratory of Structural Virology at CETEC Masaryk University, is focused on structural analysis of viruses using cryo-electron microscopy. Did his PhD at the University of Chemistry and Technology in Prague in 2014, focusing on microbiology, and it was done using electron microscopy and atomic force microscopy on virus assembly. And he did some research stays at the University of California, Irvine, with Professor McPherson. And Tibor is really an expert now in I would say, structural virology, and he'll be talking today about the structure of tick-borne encephalitis virus immature particles solved by cryo-electron microscopy and subtomogram averaging. And with this, Tibor, the floor is yours. Good.
Okay. Very warm welcome from Brno, CETEC Brno. This presentation will be about structure of immature particles of tick-borne encephalitis virus solved by cryo-electron microscopy and subtomogram averaging. So what tick-borne encephalitis virus is, it's a flavirus, similarly like dengue and West Nile virus or yellow fever virus, which you maybe know better. It's an RNA plus genome virus, which is enveloped by a lipid membrane. So it belongs to envelope viruses. And because it's tick-borne, of course, it's transmitted mainly by ticks. It's spread almost all over the world, mainly in Europe or the East Asia. And it causes several severe diseases like meningitis and encephalitis. So this is the reason why it's interesting and important to study tick-borne encephalitis virus also at the structural level. If you go to the life cycle of tick-borne encephalitis virus, we can split as usual for the viruses for the early steps, where the virus attaches to the cell, enters the cell, and relieves the genome. At this stage, the virus is called so-called mature virus because it's infectious and can infect. The structure of this mature virus was already solved by us in 2018. And on the left, what you can see is a micrograph of tick-borne encephalitis virus prep, visualized using cryo-electron microscopy, where the beautiful round particles that you can see here. A second, I will switch on this. These beautiful particles that you can see here, the roundish ones are the mature particles. They are sometimes a little bit irregular, but mainly regular, and those are very easily reconstructible using cryo-electron microscopy. And then you can see a cross-slice through reconstruction, where you can see that this virus has a double membrane, which is inherited from the cell during the budding process, which is densely covered by the so-called envelope proteins, the ectodermis of the envelope protein, which is anchored inside the membrane. Inside the membrane, there is another protein. It's called the M protein, the membrane protein, which is forming a complex with the E protein. The molecular structure looks as follows. The surface is densely covered by the E proteins. All the E proteins are essentially the same, however, they are forming different interactions. Like here at the 5-fold axis, they are forming pentameric interactions, here at the 3-fold axis, trimeric interactions, and here at the 2-fold axis, dimeric interactions. However, if we take a closer look, we see that the asymmetric unit is composed of a dimer of an E protein, and underneath this dimer is another dimer of M protein, which together forms a tetramer, plus a half of another dimer of the E protein above it. We can see that the bottom part of this is anchored in the membrane. In the ribo representation, we can see that we have E proteins colored in colored in blue, red, and green. And at the very tip of the E protein is a very interesting part here. This is called the fusogenic loop. The fusogenic loop is an important part of the E protein because during the entrance of the virus and the release after acidification inside the endosome, this fusogenic loop is fusing the membrane of the virus with the membrane of the endosome, and then it's ejecting the genome, the RNA genome, from the inside of the particle. At the bottom, you can see how the E protein is anchored inside the membrane. The membrane is depicted by this magenta dashed line. And there we have perimembrane and transmembrane helices, as well as there is the M protein, which is here in light green and orange. And this M protein is totally covered by the E protein. So from the outside of the particle, the M protein itself is not visible at all. Now, this was the early stages. We can see that the ejection of the RNA takes place at the lower pH of 6.0. And now for the particle, when it starts to assemble inside the cell, which starts at the membrane of the ER, it's important when this particle will be transported from the near neutral pH of the ER 7.2 through the Golgi apparatus, where the pH will be lowered, that it won't fuse with the membranes of the cell. So therefore, the virus creates a so-called immature form, which is not able to fuse with the membranes. And this immature form is maturated by a cleavage of protein, of PRM protein, which is covering the E protein of the particle and preventing the fusion. And later, when the particle buds out from the cell, the PR protein dissociates. PRM protein dissociates from the particle and forms a major virus. Now, what we did is that with NH4Cl, we neutralized the trans-Golgi apparatus of the cell, and therefore there was no cleavage of the PRM protein. It remained associated with the virus, and we tried to solve the structure of the immature particles. This part was quite tricky to do, mainly the purification of the virus, because it's a BSL-3 virus. So you have to do the purification of the BSL-3 laboratory, and then after fixation, you can work with it on the microscopes. So first we did it with single particle analysis. We collected several thousands of micrographs. You can see that these particles are not anymore nicely smooth. These particles are more rough and fluffy. However, in the prep, we have also a little bit of nature particles. And when we average these particles, we get this beautiful particle that looks nice, spiky, as from the micrographs. And it went down to 7.1 angstrom resolution, which is not bad. The so far published particles, immature particles of dengue virus, Zika virus, West Nile virus are all around 8 angstroms. So this is very close to that, a little bit better, but still it was a bit too far of what we wanted to have to solve the structure and the interaction of the E protein with the PRM domain. So what we did is that we just extracted from the micrographs the positions, the coordinates of the subparticles that are covering a single spike out from the surface, and therefore every particle will contain 60 of these single spikes. And we did subparticle averaging. Using subparticle averaging, we were able to reach a 4 angstrom resolution, which is not bad, and I would say already a buildable. It's already a buildable map. 4 years ago, we had a very similar resolution, the structure of this of the mature virus. So, or we can see that this map consists of not only the E proteins, but also the M protein that is sitting on the top of the E protein. So with this lighter, you can see the PRM domains that are covering the top of the E protein. If you go to ribbon representation, again, we can see that we have in dark colors the E protein anchored in the membrane. And then in lighter colors, we have the PRM part, which is covering the top of the E protein. And this forms a kind of spike. So far we had for the mature particles, we have very smooth surface. This way we have a spiky surface of the immature particles. Furthermore, the PRM is connected to the M protein. However, we don't see the full link of this connection. And after the cleavage with the furin, which was in this case prevented, then at the neutral pH, this PRM part is falling off and the E protein forms the mature particles. What's important here is that you can see the fusogenic loop that is covered by the PRM, which is preventing the interaction of the virus with the membranes. So here you can see it better on the molecular surface representation that the fusogenic loop depicted in magenta is fully covered by the PRM, and the PRM is holding the E protein in a conformation that you have a kind of dimer of E protein and PRM, plus another, let's say, kind of orphan E protein PRM that is attached from the side to it. So this way, this is a trimer, but this is a non-symmetrical trimer. So we cannot reconstruct this in any symmetry like C3. We have to go down with C1 symmetry. As well, we can nicely see that in the membrane part, here in the membrane part, we have the membrane helices of the M protein, or the part of the PRM protein, which is in the membrane, and the membrane part of the E protein, which is consisting with transmembrane helices and perimembrane helices. If we compare on the left, the mature form, and on the right, the immature form, we can see that obviously the PRM is sitting on the top of the E protein and protein in the fusogenic loop, but also there is a major change in the conformation of the E protein. While it looks very similar, during the maturation process, there is a flip that flips down the E protein towards the surface of the membrane, which goes like this. And it sits on the membrane. And another thing which is very important is a major change in the helices of the M protein and the E protein that are located inside the membrane. These helices have a different relative conformation to each other, so the helices needs to be rotated and a little bit translated towards the E protein transmembrane helices. As well, a little bit the perimembrane helices have a slightly different conformation in these two particles. Because of this, because the difference of the membrane helices or the localization of the membrane helices in the mature and the immature form of the particle, we can see another effect. The effect is that the membrane underneath the E protein looks a little bit different in the mature form of the TBEV than the immature form of the TBEV. In the mature form, you can see that the membrane is nicely curved and ringed from the inside, whereas in the immature form, it forms more like a circular surface. And this is caused by the tension that these helices are making in relative to each positions. Now, this was all single particle analysis, and I promised that there will be a little bit of tomography also, or mainly I was promising that there will be tomography here. So we tried several times tomography of the ITBEV, and usually we were limited by the amount of the immature particles, because for single particle analysis, it's a little bit easier to collect the data. So you can collect thousands and thousands of micrograms, and you have a low concentration of particles, then you just collect more data. and you can do the averaging. However, for the tilt series takes a lot longer, and you need a very good quality. So we were able to have a very nice, decent concentration of immature particles, and we did tomography of the sample. We collected 193 tilt series, and this we reconstructed into tomograms, where we can see nicely that we have nicely visible immature particles, some cellular parts that are coperificants from the cell, as well a little bit of mature particles. The data set was collected a little bit higher epics than for the single particle. Nevertheless, we haven't reached the Nyquist of this data set. What we did next, when we collected our tomograms, we performed template matching. So what we used was a single spike from a single particle reconstruction, a little bit surrounded with other spikes to make the correlation easier and more signal for that. However, for the sub-tomogram averaging itself, we used only the central single spike. So if you do template matching on a tomogram like this, and we have 193 of these, then you can locate the positions and the initial orientations of these spikes. So everywhere where we found the correlation, we put a dot, and we can color these dots according to the correlation coefficient. And this way we had something around 200,000 subtomograms, and we can average them together. What we did, we averaged it, and we reached a 4.7 angstrom resolution, which is not that great, but very close to the one that we have from the SPA. You can see that the density map looks very similar. If we compare it with a single particle with the 4 angstrom resolution map, it looks essentially the same. Still, single particle analysis has a little bit more detail, so this map is a little bit more featureful. But if we have this, now the question is what we can do with our tomograms. With our tomograms, there are multiple things that we can do. Now that we have refined the positions and the orientations of our subparticles, then we can virtually place back these subvolumes into a tomogram and create some virtual volume where we have perfectly aligned subtomograms of our spikes, and then we can analyze these particles. Out from this, we can see that our particles are greatly heterogeneous. First, we can see on the right that the particles are incomplete. They always have some kind of holes on the side. As well, the particles are not exactly spherical. This is colored by from the center by the radius, and you can see on the top it's a little bit darker red than it's on the bottom of the particle. That means that the particle is a little bit squished. Now you may argue that the particles are incomplete because they are touching the air-water interface. That's why we can show it in these placeback particles that this is not the case, because on the top and the bottom of many of the particles, we can see that the particles are nicely covered with the subparticle subtomograms, and therefore not the air-water interface or not the particles that are touching the air-water interface is causing these irregularities. Another thing that we can do is we can register the positions of the refined positions of the subparticles, and then we can cluster them into the cluster of particles. So to assign every subparticle, subtomogram to one particle, and then find the center of the particle and just fit a sphere over it. And because now we have a sphere, we can see that what subtomogram belongs to which particle. And this we can plot out, for example, how many subtomograms we have per particle, or we can measure the particle radius using this subtomogram averaged particles, or sphere fitted averaged particles, where we can see that the subtomograms per particles greatly vary, while the particle radius is quite constant. So the particle radius is around 300 angstrom, which goes to 60 nanometer in diameter. This is what we see from our micrographs. Conclusion, well, we can see that if we go for a perfect immature particle that we assemble from our molecular model, we can see it's nicely spiky and its organization greatly differs from the mature particle. And during the maturation process, all these proteins where the PRM is dissociating has to undergo a shift and rotation to achieve the final positions on the particle. Of course, this was not done only by me, but many of my colleagues helped me. My boss, Pavel Plevka, his group, Lenka Šmerdová did the wet lab work in the BSL-3 laboratory. Originally, we started with a group with Daniel Růžek. They also provided a lot of useful samples for that, and the cryo core facility of the CETEC, where I really have to recommend Jiří Nováček and Michal Lobiak, who recently left, but they are very helpful, and we were able to collect all the data with state-of-art microscopes and detectors. So the data was collected on cryos and the K3 detector, and for the tomography, we use this new base tomo scripts that are greatly speeding up the acquisition process. Of course, there's no science without any funding, without any money, so you can see all the icons of the funding agencies. And with this, I would thank for your attention.
Great. Thank you, Tibor, for the great talk. I'm looking for questions, but I cannot see any questions right now in the Q&A. I have a question for you. So the immaterial particles, they are mostly incomplete, or all of them incomplete in your observations?
Yes.
So is that, yeah? Yeah.
I guess that the question will be about if this is natural or not natural. So when we average the major particles, we were able to get particles that were very nicely averaging to down to 4 angstrom resolution, and now we have even better resolution from them. And that says us that these particles are complete. Whereas the immature particles always seems to be incomplete, or the major part of them is incomplete. We cannot rule out that this is because of the purification process itself. So maybe during the purification process, or this when you are neutralizing the Golgi apparatus, you are also affecting how the viruses are produced inside the cell, and the cells are then forming these incomplete particles. We don't know if this is the main cause or there's some natural reason for that. This we cannot rule out. Yeah.
Okay. So is it could it be related somehow to the content of the particles? I mean, they were mostly empty. Is that right? There's nothing in there really.
Those are not empty. The immature particles are full as well. So this was mainly maybe caused on the image by the normalization, but the immature particles are genome containing as well. And the one that was placed back on the subtomogram averaging, there we placed back the volumes that had masked out the genome. So that's why it looked like just a shell of the spikes. But actually the genome, the average genome was masked out.
Mm-hmm. Yeah, okay, understand. I still cannot see any question in Q&A.
Someone? Yes.
Okay.
Hello, I had a question, so I unmuted myself. Hi, this is Maria Nassasna speaking from Helsinki. Hi, Tibor, thank you. Very, very interesting talk. I have one question that it's about the subtomogram averaging. So you have the plot where you mapped the subtomogram numbers per particle.
Yes.
And there were some counts which exceeded 60.
60.
Yeah, could you elaborate?
This was the question I was waiting for, actually. If somebody noticed that there were some subtomograms that had 60, there are only a few of them. And this is because how we did the clustering of the particles was done automatically so far. So there was no hand curation of this. And sometimes what's happening is that if we maybe go back, able to go back, we can see some parasite particles, for example, here, that there's an extra particle that's obviously not on the particle surface. So there was something mistemplate matched, and also it was not classified out during the refinement process. That's one cause of it, that there are more than 60 of them. And another cause is that when we are doing this clustering, we are doing this recursively, that we are looking for the neighboring particles that are close enough. So we are defining some 130 angstrom distance between the particles that are considered to be related, to be neighbors. And if you have two of the particles that are closer than this limit, then these particles are kind of grouped together. And this always, we have some tomograms where you have something around 100 because you have two particles that are clustered together. This has to be fixed by hand. So we will need to hand curate these particles that were wrongly clustered. And this is the reason why you have a few of particles that have more than 60 counts in one or tens. You have more than 60 subtomograms. The interesting particles for us are these that has 60 that are close to complete ones, or should be virtually complete.
Yeah, I see. Thank you. Yeah, that's very clear. Thanks very much.
Good. Okay. Is there any other question from the audience? I actually have one more question, maybe a tiny one. You mentioned the fusogenic loop. Yes. Is it expected that the loop or the protein it's sitting on would be actually somehow changing its conformation again when it's actually attacking the cell and it's making the fusion with the cell surface?
Yes, exactly. So there's a third conformation of this E protein. When it enters the cell and the pH is lowered, then from these dimers that are sitting on the surface of the particle, it's actually converting into trimers, and these trimers are making a triangular shaped spike that has three of the fusogenic loops at the tip of this spike, and this spike is actually attacking the membrane and making the fusion with the membrane. So this is another conformation. This conformation was crystallized so far. It's very hard to study it in vitro because if you lower the pH with the mature particles, then these particles start to fuse with each other in all the membranes, and it just makes a huge clumps of aggregates, and those are not visible on the microscope. It's completely covering the grate. So far what they did is that they, the other groups were crystallizing it at low pH, and they got a trimeric structure that has a tip, which is formed by three of the fusogenic loops.
Yeah, okay, clear. Very nice. So thank you, Tibor, again for your talk and for answering the questions. And I'll move over to the next speaker. I'll try. Could you, yes, please stop sharing your screen.
Just a second.
Okay, I'll try do my intro. There we go. So the next speaker is Ondřej Vaněk from the Faculty of Science, Charles University in Prague. Ondřej did his PhD in biochemistry at Charles University, focusing on structural studies of natural killer cell receptors. He had two postdoc stays in Vienna Biocenter and the Technical University of Vienna, looking at biophysical methods for protein interactions and single molecule localization microscopy. And he's currently the head of the Laboratory of Structural Biochemistry of Immune Recognition at the Department of Biochemistry, Faculty of Science, Charles University. I have collaborated with Ondřej, and I know he's been working a lot in the area of structural biology of immune recognition, NK cell receptors, and also recently he's involved in the design of immunoactive therapeutic protein constructs, as it says here. So today, Ondřej will be talking on the topic of the human receptor ligand pair, NKRP1-LLT1, and what it brought from the crystal structure about the clustering of these receptors and how microscopy helped in actually discovering what is happening in the immune synapse. So with this, I stop sharing and invite Ondřej to speak.
Yeah, so thanks. Thank you, Jan, for the introduction, and thanks the Instruct for the opportunity to for giving me the opportunity to present our work today. And I hope you can see now my presentation.
Yeah, it works.
So perfect, thank you. So basically, I will present you the work and story behind a paper that has this name, and the authors are listed. In my presentation, I will give you a brief introduction into NK cells and their receptors, and then I will switch to the two of our interest for today and how we struggled with producing them, characterizing, and then solving the crystal structure, and then what we learned from that, combining structural data with solution biophysical data and observation from cell surface using super resolution microscopy, and then functional assays to basically verify or answer the question whether what we observed in the crystal structure is real or not. Okay, so natural killer cells are lymphocytes that are bridging our innate and adaptive immunity. So they are closely related to B cells and T cells, but they lack B or T cell receptor. Instead, they possess various other receptors, and that makes them more akin to the innate immunity. And they serve various roles, important regulatory functions, and crucial role in anti-tumor and also antiviral immunity. Concerning the receptors on their surface, there are many. I will not describe all of them, just to mention that there are activating and inhibitory and various other chemotactic adhesion cytokine. And basically what all these receptors do together is that they are trying to assess whether the other cells in our body are healthy or, for example, tumor or virally infected or stressed, damaged cells. And they do so by interactions of these surface receptors on NK cell with other receptors that are sending either inhibitory signal or activation signal. So we can have tolerance where in case when the markers of good health are present, or activation leading to cytotoxic reaction and killing the target cell when the markers of good health are not present and the activation signal overcomes, like in this stress-induced case, for example. So structurally, all these NK cell receptors can be divided into two big families, immunoglobulin-like and C-type lectin-like, and we are studying both, but today we will focus in my talk on this one, and especially on NKRP1, LLT1. I will introduce them shortly. What's interesting about this family, C-type lectin-like, is that in the genome, and this is true for both like rodent genome, mouse or human, the genes for receptors and their ligands are intermingled. They are co-inherited, so there is a conservation, evolutionary conservation of these genes. And this picture is showing, it's like a textbook picture, yeah, more maybe 10 years ago, showing various pairs of these receptors and ligands, and it's just a scheme. So others, and we as well, were interested in how does it look in real life. So then structures of various complexes were solved, so this picture became a bit more clear. But typically you see like a monomer or monomeric complex in the structure. So this is really in this drawing. So it's based on the observation in the crystal, and the question is how is it really on the cell membrane. So for example, this receptor ligand complex, which we are also studying, but I will not go into detail about this today, is also oligomerizing, forming bigger clusters on the cell surface, which is important for the signaling. And this is exactly what we are interested in. So coming back to our two proteins of interest for today, NKRP1 is a human NKT cell receptor. It has inhibitory function, and it's basically the NKRP1 family was the first natural killer cell receptor protein discovered. It is was and is used to describe NK cell as a marker, and until recently the structure was not known, and its ligand, called lectin-like transcript 1 or LLT1, is expressed on other immune cells, also NK cells, but also other immune cells, and it has regulatory function. So this interaction typically leads to suppression of NK cell activity, regulatory function, but in many cancers, many cases of various cancers, including glioblastoma, but there's more. LLT1 is unfortunately overexpressed by those cells, and then it inhibits our immune response. So when we started to when we wanted to study these two proteins, so the structure and the complex, we needed to produce them in a soluble form. So naturally, both proteins are transmembrane. LLT1 has intracellular region, transmembrane, and extracellular domain. It should form a disulfuric dimer. And the ectodomain, the binding competent domain, is possess 5 cysteine residues. And when we align similar sequences, we noticed that one of the six conserved cysteine residues is missing. So in this position 167 in wild type is replaced by histidine. And when we expressed in human embryonic 293 cell line, the ectodomain of LLT1, we observed a lot of aggregation, a lot of misfolding, and by using mass spectrometry, we learned that there's sorry, this is here. So the wild type has misconnected disulfides due to the presence of these odd cysteine residues. So we created 2 mutants. In one, we mutated the odd cysteine to serine, but this was not expressed at all. In the other, we retrospectively mutated this histidine to the cysteine residue, which restored the missing disulfide bond, and that lead to very nice stable production. So this is this blue chromatogram. So our take-home message for ourselves was that in our protein constructs, we should take care and mate our cysteine residues. Another important step was to learn how to produce these proteins in 293 cell line, and especially taking care about the proper glycosylation profile to enable crystallization and structural studies. So we, with help from basically Instruct, I will be mentioning Instruct quite a lot during the presentation because it really helped during the work and over the years. Mostly it was collaboration with OPPF in Oxford. So it was here where we initially learned how to use this expression system, and also that the 293S N-acetylglucosaminoyl transferase 1 cell line, which is deficient in this enzyme. So all the N-glycans on proteins produced in this cell line are this type, uniform and cleavable. Is very helpful for making proteins that are crystallizable. And you can appreciate that by looking on this SDS page, where you will see LLT1 wild type forming these unwanted misfolded forms. Then the mutant produced this wild type glycosylation. You see these smears, but then when we produce it with uniform glycosylation, we see nice 2 glycoforms, 1 glycan, 2 glycan, which could be deglycosylated to very, very nice protein that we then crystallized and solved the structure that you can see here. It's a non-covalent homodimer. We could observe all the 3 disulfide bonds, including this one, which was reconstituted by the mutation. And yeah, basically, otherwise the structure follow a bit of an expected fold. About the time we published this, months later, 2 months later, another paper came out describing the same structure from a Japanese group of Katsumi Mayanaka. And then they also proposed a model for the structure of the complex based on some other structures and some interaction data. I will show you that the reality is different from this model. And to move towards the structure of the complex, we needed to produce the receptor human NKRP1, which was a challenging task. So originally, years ago, we were also using E. coli. That leads to inclusion bodies, refolding not successful. When we tried the expression in hex cells, the yield was very low. So with the help from Instruct and OPPF, we created a library of 46 constructs differing in the length of the stalk region, trying different solubilization texts and whatever. And basically high throughput expression screening was performed and in OPPF, but there was no improvement. In E. coli, we really were not able to obtain soluble proteins. So the library was then recloned to vectors enabling insect cell production using baculoviruses, which was not scalable. So again, no success. And then the same library was screened in our hands in hex cells. And basically, we learned that the original construct was still basically the best one. So what now? We have the right construct and expression system, only the yield is too low. So we didn't give up. And it was the work and effort of my former PhD student, Jan Blaha. So I would like to credit him here, because it's really most of the significant part of the work I'm showing today is really thanks to his efforts and courage. And this critical step that we needed to do in our mind basically was to switch from doing transient transfection in HEC 293 cells to stable transfection. So again, we use the vectors from OPPF, and we created a stable polyclonal cell line that, to our surprise, secreted NKRP1 in very good yields. Later, and this is what we do today, we switched to piggyback system for making stable polyclonal cell lines that are even inducible, and the yields are even better. So suddenly it was not a problem to obtain very nice protein and to crystallize it. So we obtained these nice crystals with help from OPPF, and Karel Haros collected the data sets at Diamond synchrotron, and we saw the structure of NKRP1 alone, but to our delight, also the complex. And here I have to credit, well, our chair for today, Jan Dohnálek and his team in BioSef, and especially Teresa Skalova, who was the leader of the structure solution, refinement, and all this part of the work, crystallography work. So coming back to the to the structures, what we have learned. So on this figure, you see the structure of NKRP1 monomer and then the dimer. So this receptor is also forming a dimer. It should on the cell membrane, but with our soluble ectodomain, we were expecting that it could form maybe non-covalent dimer, although in solution we always see just the monomer by various techniques. I will show you as well later. So this non-covalent dimer is exactly the same when we compared the structure of glycosylated, deglycosylated, and deglycosylated in a complex. But what was interesting was that when we compared the structure of non-covalent dimer of the ligand, LLT1, and the receptor. So if you look down here on this bottom right, here LLT1 and NKRP1 proteins are aligned using the monomer in one of the two monomers in each protein. So you see that the fold of the monomers is very similar, but the way how the dimers are formed is opposite. So we colored here helix alpha 1 in red and helix alpha 2 in yellow. In one case, the dimerization interface is based on helix alpha 2, and in the other on helix alpha 1. And that's quite important, and that's what's different from the previous models of how this NKRP1 protein could look like and how the complex could look like. And because of that, I will come shortly to the complex. You will see how it can cluster on the cell surface as well. So, but still, what we have learned from the structure of the receptor itself, when we examined the dimerization interface closely, we noticed several interesting points. So, for example, here there was a publication in 2015 describing single nucleotide polymorphism causing mutation of isoleucine 168 to threonine residue. So it's here the authors propose that maybe it's at the binding interface. Actually, it's not at the binding interface, it's on the dimerization interface. So it could, the shift from the hydrophobic amino acid to more polar amino acid is disrupting, at least partially, the dimerization, which is then translated to the overall to weaker binding overall affinity or avidity and weaker outcome of the signaling, which which is what they observe. And also glycosylation is present at the dimerization interface, which you can see here. So when we look on this part of the dimerization interface from the top, you see that the N-glycans, and we can only see the initial N-acetylglucosamine, but the glycan is of course bigger. The glycans are touching the other monomer unit in the dimer and are contributing by making hydrogen bond contacts to the dimer formation. And then we also supported this by making a mutant in this glycosylation site, and then we really see that glycosylation state in this position is affecting how the protein forms the dimer. Moving towards the complex and how we characterize it. So this is the structure of the complex, crystal structure of the complex. We have the non-covalent dimer of LLT1 in contact with two non-covalent dimers of NKRP1, and there is one symmetrically related NKRP1 dimer as well. The two binding interface are different. The structure is not symmetric. NKRP1 uses pretty similar set of amino acid residues that are touching LLT1 in this type of contact, whereas LLT1 uses or is being touched by NKRP1 on slightly different positions, so different set of amino acids here and here. So we call it primary interaction mode because this is the one that was kind of expected based on other previous observation from other studies on related complexes, and secondary interaction mode. And when we looked in the crystal how the molecules are arranged next to each other, we saw this chain-like formation. So in this figure, NKRP1 molecules are in magenta and cyan, and green, red, yellow is LLT1, and basically a dimer of NKRP1, two dimers of NKRP1 are bridged by one dimer of LLT1, and so on and so forth. So the question was, is this kind of chain-like oligomer formation just something that we can see in the crystal based on the, how the crystal was formed, or could it actually happen on the cell surface? Could this be the way how the two proteins overcome their relatively low affinity? So the affinity for like monomer-monomer interaction is only about 50 to 100 micromolar, depending on the method you use and how you observe it, but it's rather weak interaction. So could it be that this is the way? So this was like the hypothesis that we wanted to test, that we have this dimeric NKRP1, we have this dimeric LLT1, and this model was the standard model that was proposed until now or recently, and this was the new model. So could it be that actually this is what happens on the cell surface? So avidity instead of affinity. So we characterize this using various biophysical methods, starting with the methods in solution, like analytical ultracentrifugation, also microscale thermophoresis. So for example, here you can see sedimentation profiles of NKRP1 and LLT1 and their mixture, 1-to-1 mole ratio, seeing how the complex is formed when we go up with the concentration. Well, and then this is not the case when we use a mutant. So we created a mutant. I have to go back. So to explain this properly, we create a secondary mode interaction mutant, abbreviated SIM. So SIM, LLT1 SIM, is LLT1 mutated in these residues. So the secondary interaction interface is not working, but we still preserve the interaction in the primary mode, which we can prove, for example, which we can prove by these methods basically here. So we still see interaction, we still see binding and complex formation with the mutant, but the particle, the complex that is created is smaller, and the interaction has a weaker affinity. And I was able to globally fit these biophysical data. And the question was, so is it like A to B interaction, or do we have a dimer interacting with a monomer, like BAB interaction? And basically, based on this, we were able to confirm that really the wild type is forming this expected complex, where we have on a dimer of LLT1 two monomers of NKRP1, whereas for the mutant we only see dimer of LLT1 with a monomer of NKRP1 because the other interaction interface is mutated. Then we tried to observe this also by using small angle X-ray scattering coupled to size exclusion chromatography. So these are SETSAX data collected on diamond synchrotron beamline. The main peak is composed of the main complex or its various states, but we were happy to see this small peak preceding the main peak. The chromatography was performed at the highest concentration we were able to achieve, I think like 18 mg per ml. So in this small complex was a sign that even in the solution we have some higher order complexes or higher order arrangements. We tried to analyze those data, and because we knew it's a mixture of various particles, we were not modeling these data by one or the other possible arrangement. But what Jan Blaha did is that he created a library of all theoretical possible combinations of dimers of LLT1 and NKRP1 in primary mode, secondary mode, both of them, just one, just the other. And then we used an algorithm called oligomer, which allows fitting SAX data by using this library of structures. So it was like, I don't know, 40 structures. And the algorithm is trying to find the best combination of all these different structures explaining the observed data. And then so some of these frames and curves are shown, and the values are shown here. So basically what it brought to us is that the algorithm, without our wishful thinking, selected or preferred the theoretical arrangements that were composed of dimers of LLT1 bridged by dimers of NKRP1 in alternating primary and secondary mode. So the same arrangement that we see in the crystal structure, and not the other theoretically possible arrangements based on just one or just the other interaction mode. So it was supporting the data. But then, okay, is it really the case on the cell surface? So we performed super resolution microscopy of this system using D-Storm microscopy method with cluster visu software to process the data using Voronoi tessellation. I have to credit my former PhD student Barbara Kalouskova, because she was the leader of this part of work. So what we have learned from here, we were comparing super resolution microscopy data for the NKRP1 alone, or so cells expressing NKRP1, and then those cells in presence of LLT1 in high concentration in solution, and it was either LLT1 like wild type or the secondary interface mutant. And we monitored the changes in clustering parameters, and based on these data, we can confirm that there are statistically significant differences in cluster size for NKRP1 when LLT1 is present, but not when the secondary interface mutant is present. So this basically confirmed the clustering or this that this higher order complexes are also present on the cell surface. And then the next question was, is it really relevant to the cell function. So we performed NK cell cytotoxicity tests, meaning you have NK cells freshly isolated from the blood of healthy donors, and as a target cell, a leukemic cell line, which you incubate with NK cells. And when we do not add anything, most of the target cells are being killed. But when we add LLT1, and we have like 2 different concentrations here, this causes inhibition because NKRP1 is an inhibitory receptor. So we observe inhibition of this killing activity of NK cells. But when we, instead of LLT1, use the secondary interface mutant, you see the difference in the effect. So that proves that the secondary binding interface on LLT1 is really functionally relevant, and that this higher order complex formation is really important for the clustering and the function of this receptor. So for this cytotoxicity, I need to credit Dennis and also former student of myself. And then basically all the work is then published now in this nice publication in Nature Communications from last year. And that's basically the end. Maybe before I go to the really Thanksgiving, I should summarize that by now we also have structural data and some other supporting data for other complexes of NK cells, showing that this clustering is not just for this one particular complex, but it's quite probably a strategy that is more general for at least C-type lectin-like NK cell receptors, and that could be maybe explored for some therapeutic construct design and then. So I would like to credit many students, of course, that helped, and collaborators, Teresa, Jan, Karl, Yuang, some funding, last but not least from Instruct-ERIC, including also the R&D pilot project EVERT. And I think I'm late with time, so that's all from me. Thank you for your attention, and I'm open to questions.
Thank you, Andre. Thanks a lot for this interesting and complex story. Are there any questions from the audience? I cannot see anything in Q&A. Is there anybody who would like to just shout and ask a question?
Maybe I would have a question that I guess this receptor downstream is activating some kinases and through the membrane, and this alternative binding is activating these kinases the same way, do you think? Or will it be efficient the same way, more or less? Do you have some data for this?
That's a good question. We do not have data for this. You are right. There are there is an ITIM motif, so inhibition immunoreceptor tyrosine inhibition motif in the intracellular part of NKRP1, and then the signal is transmitted to the cell. It's not that easy for NKRP1 to really observe this biochemically. We were considering it, but in the time that we had for this study, we were not able to look into this. But coming back to these functional data, we can concentration, let's say, of these receptor molecules on the cell surface, then leads to like triggering the downstream signalization. Whereas if this is not happening, if they remain scattered alone, it's not happening.
Thank you.
Thank you, Andre. Do you think I guess that what is actually happening on the interface and then inside the cell, that cryo-EM tomography should help in the future to really have a proper look? Do you know if anybody is working on that? Using cryo tomography?
I'm not aware of that. Maybe the size of these proteins could be a bit limiting. I don't know what is the threshold nowadays. So it's about 30 kilodalton for monomer, 60 per dimer. So if this is doable, I'm more than happy to to work on this with someone who's expert in cryo-EM tomography.
I guess labeling would be really necessary here. I mean, they are tiny. Yes, they are tiny. But
for another complex that I mentioned in the beginning, NKP30B7H6, where the receptor itself is forming oligomers, we are following that by cryo-EM single particle. And to increase the resolution, we plan to use nanobodies in form of megabodies to help that. So maybe this would be the approach here as well.
Yeah. Okay. We cannot see any other question. So if there is another question, you have the last chance now, right now. And that's not the case, I guess. So thank you, Andre, and thank you, Tibor, again for joining us for this Instruct webinar.
Thank you.
Thank you for your results and your willingness to share with us. And thanks everybody for joining the webinar and listening in. And I hope we can meet at the next Instruct webinar, I guess. Yeah. Thank you. Thank you and bye-bye.
Thank you. Have a nice day.
Thank you.